Welcome to SCT 106 Biochemistry 1. This session encompasses the sources of carbohydrates and the introduction of monosaccharides in terms of carbon numbers and their classification as aldoses and ketoses, the different types of isomers, open chain and ring chain forms, derivatives of monosaccharides and their functions, the glycosidic bond, structure and functions of common disaccharides, oligosaccharides, and polysaccharides, and finally, the structure of cellulose and amylose. Carbohydrates are one of the most abundant biomolecules on Earth. Through photosynthesis, tons of carbon dioxide and water are converted into cellulose and other plant products. Certain carbohydrates, such as sugar and starch, are a dietary staple in most parts of the world, and the oxidation of carbohydrates is the central energy yielding pathway in most non-photosynthetic cells. Insoluble carbohydrate polymers serve as structural and protective elements in the cell walls of bacteria and plants and in the connective tissues of animals. Other carbohydrate polymers lubricate skeletal joints and participate in recognition and adhesion between cells. Carbohydrates are polyhydroxyaldehydes or ketones, as we shall see later throughout the session. Note that in addition to carbon, hydrogen, and oxygen, some carbohydrates also contain elements such as nitrogen, phosphorus, or sulfur. There are three major classes of carbohydrates, and these are monosaccharides, oligosaccharides, and polysaccharides. Monosaccharides, or simple sugars, consist of a single polyhydroxyaldehyde or ketone unit. The most abundant monosaccharide in nature is the 6-carbon sugar glucose, sometimes referred to as dextrose. Monosaccharides of more than 4 carbons tend to have cyclic structures. Oligosaccharides consist of short chains of monosaccharide units or residues joined by characteristic linkages called glycosidic bonds, which we shall see later. The most abundant are the disaccharides with 2 monosaccharide units. Typical is sucrose or cane sugar, which consists of the 6-carbon sugars, glucose, and fructose. All common monosaccharides and disaccharides have names ending with the suffix -ose, O-S-E. So as you see, for instance, glucose ends with the suffix OSE. So is fructose, so is sucrose, etc., etc. Now, in cells, most oligosaccharides consisting of 3 or more units do not occur as free entities but are joined to non-sugar molecules in glycoconjugates. These complex carbohydrate polymers are covalently attached to proteins or lipids and act as signals that determine the intracellular location or metabolic fate of these hybrid molecules called glycoconjugates. Polysaccharides are sugar polymers containing more than 20 monosaccharide units, and some have hundreds or thousands of units. Some polysaccharides, such as cellulose, are linear chains. Others, such as glycogen, are branched. Both glycogen and cellulose consist of recurring units of glucose, but they differ in the type of glycosidic linkage and consequently have strikingly different properties and biological rules. Now, monosaccharides are generally soluble. They are soluble in water but insoluble in non-polar solvents, and most of them have a sweet taste. There are two main classes of monosaccharides, being aldoses and ketoses. Aldoses have the aldehyde group in them. So for instance, you have glyceraldehyde having the aldehyde group C double bond OH at the end of the chain. You have the C double bond O, which is the aldehyde group in glyceraldehyde. So you see the aldehyde group also in glucose. You see same in ribose, and you see same in deoxyribose. These monosaccharides with the aldehyde group in them are called aldoses. Now, the second group, ketoses, have the ketone group, the C double bond O group along the chain, not at the end. The ketone group does not exist at the end of carbon chain. It exists somewhere along the chain apart from the ends of the chain, the ketone group. These monosaccharides, dihydroxyacetone containing the ketone group, the fructose also having the ketone group. These examples of monosaccharides are ketoses because they have the ketone group in them. The simplest monosaccharides are trioses, and these trioses contain 3 carbons. So glyceraldehyde is having 1, 2, 3 carbons. So is dihydroxyacetone also having 3 carbons in them. These are trioses. And so for glyceraldehyde, that is for glyceraldehyde, that is an aldotriose because it has the aldehyde group and it has 3 carbons in it, and thus it is an aldotriose. Ketotrioses will have, you have the dihydroxyacetone being a ketotriose because it contains the ketone group and it is made up of 3 carbons, 1, 2, 3, and so it is a ketotriose. Now, monosaccharides with 4, 5, 6, and 7 carbon atoms in their backbones will be called the tetroses, pentoses, hexoses, and heptoses respectively. So the most common monosaccharides in nature, glucose, is an aldose, is an aldohexose because glucose has the aldehyde group and it is made up of 6 carbons, and thus it is an aldohexose. Fructose is a ketohexose because it has the ketone group here in its structure, and it's made up of 6 carbons, and thus it is a ketohexose. Now, the aldopentoses that you find in nucleic acids and nucleotides, that is in RNA and DNA, are aldopentoses, and so you find ribose made up of 5 carbons, 1, 2, 3, 4, 5, and it's having an aldehyde group, and thus it is an aldopentose. You see same with deoxyribose, which is also an aldopentose, containing 5 carbons, and thus is a pentose, and it contains the aldehyde group, and thus it is an aldose. And these two come together to have it being classified as an aldopentose. Now, stereoisomers are mirror images of each other. So for instance, if you are looking into the mirror and you imagine yourself to be a molecule and your image that you are looking at, both yourself and your image, you are mirror images of each other. You and your image are stereoisomers. If you take yourself as the molecule and your image as an isomer of yourself, both of you are stereoisomers. Now, stereoisomers are represented with the ball and stick models. This shows the real configuration of the the atoms, the groups in the molecule. Now, there are also other ways of representing stereoisomers, and these are the Fisher projection formulas and the perspective formulas. In the Fisher projection formula, you have molecules being represented in the three-dimensional structure. The three-dimensional structure will have the horizontal bonds. So these bonds bonding to hydrogen and oxygen. These bonds are imagined to be pointing towards you as you are looking onto your screen. Your screen is a plane, and these bonds are pointing towards you, bonded to the hydrogen and the hydroxyl groups being pointed at you. And the vertical bonds are pointing out of the screen to the back of the screen, away from you. In the perspective formulas, you have the bold bonds pointing towards you, and the dash wedges, the bonds with the dash wedges are pointing away from you. Now, in representing the D and the L isomers, for instance, in the simple molecule of glyceraldehyde, if you have the hydroxyl group, as you see shaded in red, pointing to the right, this is a D isomer. If you have the hydroxyl group pointing to the left, this is an L isomer. It is the same principle in perspective formulas. The hydroxyl group here points to the right, and so this is D-glyceraldehyde. This is the D isomer of glyceraldehyde. And if the hydroxyl group is pointing to the left, this will be an L isomer. And so for any monosaccharide to be grouped as a D isomer or an L isomer, once the molecule, the structure of the molecule compared to glyceraldehyde gives you a similar configuration as glyceraldehyde D isomer, that molecule, that isomer will be the D isomer. If the isomer compared to L-glyceraldehyde is the same, similar to the configuration as L-glyceraldehyde, you have that isomer to be the L isomer of that monosaccharide. Then again, you see that you have the central carbon around which you have 4 different groups. This central carbon is called the chiral center. And to find the number of stereoisomers for any molecule, for any monosaccharide, you have the formula 2 to the power n representing the number of chiral centers in any molecule. Now, in glyceraldehyde, you have only one central asymmetrical carbon, and thus it has only one chiral center. And thus 2 to the power 1 is 2, and thus you have the aldohexose, glyceraldehyde, having only 2 stereoisomers. That is the D-glyceraldehyde and the L-glyceraldehyde. These are 2 stereoisomers. Now, here we have represented the aldoses of the trioses, the tetroses, the pentoses, and the hexoses. These are aldoses. So we have the 3 carbon, which we say is an aldotriose. You have the aldotetrose, the aldopentose, and the aldose hexoses. Now, what you need to take into consideration is you have these carbons which are in red. These are the chiral centers in each of these structures on your screen. Now, as a general rule, for aldoses, to know the number of chiral centers, you take the number of carbons that are in the structure of the aldoses. So, for instance, you have 3 carbons. If you take the number of the carbons in the aldoses as n, for you to find the number of chiral centers in the molecule, you subtract 2 from the number of carbons in the structure of the aldose. So 3 minus 2 gives you 1, and thus for this glyceraldehyde has 1 chiral center. 4 carbons, 4 minus 2 gives you 2, and thus you have the 2 chiral centers here. With the same application, you have 5 carbons, 5 minus 2, 3. You have 3 chiral centers for pentoses. Same with the hexoses, the aldohexoses. If you subtract 2 from the 6 carbons that are contained in the aldohexoses, you have 4, and thus you have 4 chiral centers in these molecules. Now, 4 and 5 carbon ketoses are named by inserting UL into the name of a corresponding aldose. So, for example, you have the 5 carbon monosaccharide aldose ribose. Now, just before the suffix ose, ose, the insertion of UL gives the name of the corresponding ketose, which is ribulose, and is the same for the aldose xylose. Once you insert UL before the suffix ose, you have the name for the corresponding ketose, which is xylulose. Then also you have ketohexoses named in relation to the sources from which they are gotten from. So, for example, fructose is gotten from the Latin word fruit, meaning the fructose meaning fruit, because fructose is gotten from fruits. Fruits are rich in fructose. So sorbose, for instance, see sorbose. Sorbose is also derived from the plant sorbus, which has berries rich in the sugar alcohol sorbitol. And sorbitol is a naturally occurring sweetener with low calorific value and is thus used in pharmaceutical products such as pharmaceutical products and sugar-free foods and oral care products such as mouth fresheners and toothpastes. Now let's turn to epimers. Epimers are two sugars that differ only in the configuration around one carbon. So for instance, if you take mannose, in comparison with glucose, you find that the difference between the structure of mannose and glucose is in the configuration of the groups around carbon 2. So you see the hydroxyl group on carbon 2 in mannose pointing to the left, whilst in glucose, the hydroxyl group is pointing to the right. All the other configurations, all the other positions of the groups around the carbon atoms are the same. So glucose and mannose are epimers. It's the same with glucose and galactose compared. You find that the difference between glucose and the galactose structure is that the hydroxyl group on glucose or on carbon 4 of glucose is pointing to the right, whilst the hydroxyl group for galactose is pointing to the left, and thus glucose and galactose are epimers. So one molecule, one monosaccharide can have more than one epimer, but always you can talk about epimers when comparing only two monosaccharides. You cannot be comparing 3 at once, 3 at the same time. You can say mannose and glucose are epimers. You can say glucose and galactose are epimers when they have difference of configuration around only one of the carbons in them. Now, thus far, we have been talking about the structure. We have been looking at the structure of monosaccharides as open chain or straight chain molecules. But in aqueous solution, aldotetroses and all monosaccharides with 5 or more carbon atoms in their backbone occur predominantly as cyclic ring structures. Now, to understand why these monosaccharides exist as cyclic ring structures, we need to understand how these ring structures are formed as a result of a general reaction between alcohols and aldehydes or ketones to form derivatives called hemiacetals or hemiketals. So if you take, for instance, the first reaction above, and you see aldehyde reacting with an alcohol, you see the hydrogen of the hydroxyl group in alcohol reducing the oxygen of the aldehyde group to a hydroxyl group, and the residue of the alcohol now have an ester-like bond with the carbon of the aldehyde group. Now, second alcohol now reduces the hydroxyl group further to have another ester-like bond with the residue of the second alcohol, releasing water, and this is a condensation reaction. Now, the formation of the cyclic rings follow similar reactions as that of the aldehyde and alcohol, forming hemiacetal. And the reaction for hemiacetal with a second alcohol giving you an acetal, and being a condensation reaction releasing water, is similar to the reaction that is between two monosaccharide units giving you giving you a disaccharide, and thus the formation of glycosidic bonds. So we will see this kind of reaction soon, where you have the aldehyde group in a monosaccharide and one of the hydroxyl groups within the same monosaccharide reacting to give a ring structure which is similar to a hemiacetal. And also in the formation of the glycosidic bonds, we will see a similar reaction where you see two monosaccharide units with hydroxyl groups also forming the glycosidic bond and releasing water, similar to the formation of acetal. You see similar also with ketones, where you have the hydrogen of the hydroxyl group reducing the ketone group, the C double bond O group here, to have a hydroxyl group on it, and the residue of the alcohol is forming an ester-like bond with the aldehyde to form a hemiketal, and a further reaction with a second alcohol giving you a ketal, where you have the second alcohol now further substituting the hydrogen of the hydroxyl group on hemiketal, forming an ester-like bond and release of water. Now, the reaction which we had just seen is now occurring intramolecular in glucose, as we see. You take the aldehyde group here and the hydroxyl group on carbon number 5. Now, if you imagine that this open chain molecule of glucose is folded to have a 6-ring structure in this way. So you have carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. What you notice is that you have the aldehyde group here on carbon number 1, and the hydroxyl group shaded, this hydroxyl group on carbon number 5, coming close to each other. Now, what you realize is that, as you noticed in the formation of the hemiacetal, you have the hydrogen of the hydroxyl group coming to reduce the C double bond O to a hydroxyl group, as you see here, reducing it to a hydroxyl group. And then there is an electrophilic attack here on carbon number 1, and you now have what you call the ester-like bond between carbon number 1 and the oxygen of the hydroxyl group, as you see in the reaction with aldehyde and alcohols, forming a hemiacetal. Now you can also see that these two molecules are stereoisomers of each other, where you have the alpha glucopyranose and the beta glucopyranose. If you take a close look, you see the H in the alpha stereoisomer is pointing, the hydroxyl group is pointing downwards in the alpha glucopyranose, while the hydroxyl group is pointing upwards in the beta glucopyranose. This way of representing stereoisomers of cyclic structures of monosaccharides is referred to as the Haworth perspective formulas. We saw the Fisher projections in the open chain, and we saw the perspective projections also. Here with the cyclic structures, we represent the stereoisomers with what we call the Haworth perspective formulas. Now, the alpha and beta anomers of glucose can interconvert in aqueous solution by a process called multirotation. And so, for instance, if you take two separate cups of solutions containing each of them having either alpha or beta, so you have one cup of solution having an alpha glucopyranose in it, whilst you have the other having only beta glucopyranose in it. Because of this process of multirotation, which occurs in solutions, after some time, these two mixtures will have the same equilibrium behavior of stereoisomerism, because you will then have in both solutions the almost equal amounts of alpha and beta stereoisomers in these solutions, because of multirotation occurring in these solutions. Now, the carbon number 1 around which you have this multirotation occurring is referred to as anomeric carbons, and thus these two molecules are referred to as anomers, just as we refer to the open chain structures. We refer to them as stereoisomers. We refer to them as the D isomer, the L isomer. In this case, these two molecules are anomers, and the carbon around which they have this rotation happening is called the anomeric carbon. Now, monosaccharides are also classified as paranoses and furanoses, and this is because, for instance, if you take glucose, the glucose ring looks similar to that of the six-membered ring compound called pyrene. If you take a close look at the ring of pyrene, this compound pyrene, there is this similarity with the ring of glucose, and thus glucose is referred to as glucopyranose, and thus it is a pyranose. So all monosaccharides having ring structures looking like the ring structure of pyrene are referred to as pyranoses. The same way you have the ring compound furan, for which its ring looks like that of the rings of fructose. For instance, you have fructose, and fructose, the structure of fructose looks very much like furan, the structure of furan, and thus is also called furanose. Now, we saw the formation of the 6-ring structure compared to the reaction of the aldehyde with the hydroxyl group or the alcohol to give you a hemiacetal. Similar reaction occurs in fructose, which is a ketohexose. And here you have the reaction occurring between the hydroxyl group on carbon number 5 and the ketone group on carbon number 2. giving you the ester-like bond, which you see forming between the alcohol residue and the alcohol residue and the carbon of the ketone group. Then, of course, you also see these hydroxyl. You see the hydrogen of the hydroxyl group on carbon number 5, now reducing the oxygen of the carbonyl group to give you a hydroxyl group. This is in a beta configuration. And of course, the carbon number 2 in the formation of the fructofuranose is the carbon number 2 is the anomeric carbon. In the formation of glucopyranosis, we saw carbon number 1 to be anomeric carbon. But in fructofuranose, you have the anomeric carbon being carbon number 2. Now, apart from the representation of monosaccharides, the six-ring structure as furanoses, or you have the six-ring structure as the paranose. You also have the six-membered paranose ring, assuming either of two chair conformations, what we call two chair conformations. Of course, in some texts, you will also see what we call the both conformations. But here we're talking only about the chair conformations, where you have the six-ring structure assuming the shape of a chair. There is, when you imagine having a six-ring and twisting it, see if you are tangling it, it forms the shape of the shape that looks similar as a chair. Now you imagine that there is this axis going through the chair. This axis going through the chair. All groups which will be pointing parallel to this axis on the carbons of these chair conformations are referred to as axial groups, and the groups which will be pointing perpendicular to this axis are referred to as the equatorial groups. Now, bulky groups on these carbons will prefer to be in the equatorial positions because there is less steric hindrance in these positions. Steric hindrance simply means the interaction with other groups on other carbon atoms. There is this interference which occurs between groups on these carbon atoms, and in equatorial positions, this interference, this steric hindrance, is less than when the groups are in the axial positions. And so, of course, when you see here on carbon number 5, you have the CH2OH, which is the carbon number 6 group, being bulkier than hydrogen, and thus you see it occupying the equatorial position, whilst you have the hydrogen on that carbon number 5, in an axial position. And you see similar things happening with derivatives of monosaccharides. So if you take derivatives of monosaccharide, which we will look at very soon, where you have some of the hydroxyl groups being replaced by other bulkier groups than the hydroxyl group, those bulky groups will prefer to occupy the equatorial positions than the axial positions. Now, two conformers are not readily interconvertible without breaking the ring. However, when the molecule is stretched and an input of about 46 kilojoules per mole is applied, this can force the interconversion of chair forms, so that though before you can convert between alpha and beta chair conformations, there has to be some breakage of bond and formation of bond. It may still happen without the breaking of bonds, just by stretching, by applying some energy up to about 46 kilojoules per mole for this to happen. Now let's go further to look at derivatives of monosaccharides, as we earlier were mentioning about bulky groups that may come into to substitute some of the hydroxyl groups on any of the carbon atoms. So if you take the parent molecule glucose here and you do a comparison, you find that in glucosamine, for instance, you have the hydroxyl group on carbon number 2 being replaced by the amine group. So you see the same thing with galactosamine. You have the amine group substituting the hydroxyl group on carbon number 2. And in monosamine as well, you have the amine group coming to replace the hydroxyl group on carbon number 2. You also see a further condensation of the amine group on carbon number 2 with an acetic group, and thus having the N-acetyl glucosamine. You see a similar in N-acetyl moramic acid, where you have the amine group also being condensed with an acetic group to give you N-acetyl moramic acid. In N-acetyl moramic acid, you also have on carbon number 3 the residue of lactic acid, in addition to the N-acetyl group, which is on carbon number 2. You see similar derivatives, similar substitutions in derivatives such as fructose and rhamnose, where you have the CH3OH, which is carbon number 6 here. If you look at the parent molecule where you have CH2OH, you now have the hydroxyl group on carbon number 6 now being replaced, being substituted by hydrogen. See that being, so you have this being CH3 reduced to CH3. Then of course you also have the phosphate. You have glucose 6-phosphate, where you have the hydroxyl group here now being substituted with this phosphate group. Then you also have the oxidation of the aldehyde group, the aldehyde group on carbon number 1 being oxidized to form a carboxylic group. Now, when you have the aldehyde group being oxidized to a carboxylic group, this is called an aldonic acid. This is an aldonic acid. If the oxidation is at the other end on carbon number 6, this will be an uronic acid. Now, you can also have an aldonic acid forming an intramolecular ester, as you see here. If you see the aldonic acid forming an intramolecular ester, and this intramolecular ester forming from an aldonic acid is referred to as lactone. Now we move to the glycosidic bond. Now we saw the reaction with the aldehyde and the alcohol forming a hemiacetal. In the formation of glycosidic bonds, you have the carbon number 1 of one of the glucose molecules having a hydroxyl group on it, the carbon number 4 of another glucose molecule having hydroxyl group on it, which behaves as an alcohol, whilst carbon number 1, as we saw in the formation of the cyclic structure, when the reaction happened between the aldehyde group on carbon number 1 and the hydroxyl group on carbon number 5 to form the cyclic structure, this structure could now be seen as a hemiacetal in that reaction, so that a further alcohol reacting with this hemiacetal should be producing an acetal. And when that happens, of course, we saw that it was a condensation reaction with the release of water. We have the release of water, the hydroxyl group, the hydrogen from this hydroxyl group, which is representing the second alcohol, combining with the hydroxyl group of the hemiacetal to give us water, and then the formation of the ester-like bond between the two monosaccharides, giving us maltose. Now the end of the disaccharide with carbon number 1 being free is referred to as the reducing end, because this end can react with copper ions and with ferric ions, and carbon number 4 on the other end is a non-reducing end because its carbon number 1 has already formed a glycosidic bond and is not available to form any to react with cupric ion or ferric ion. Now, polysaccharides are also referred to as glycans, and these can differ in their branching, or you can differ in the kinds of monosaccharide units that are in them. If you have polysaccharides that contain the same kind of monosaccharide units, you refer to them as homopolysaccharides. So for instance, if you have polysaccharide which contains only glucose units, that is a homopolysaccharide. If you have a polysaccharide that contains different kinds of monosaccharide units, so for instance, you take a polysaccharide that contains, say, glucose, and it has some you have glucose, you have fructose, you have maybe some derivatives, and acetylglucosamine. If you have such kind of polysaccharide containing different kinds of monosaccharide units, that is a heteropolysaccharide. And if you can have a homopolysaccharide that is branched or unbranched. So you will see later the branch points in polysaccharides, the bonds that give the branch points in polysaccharides. So you have the unbranched homopolysaccharide, and you have the branched homopolysaccharide. You can also have unbranched heteropolysaccharide or branched heteropolysaccharide. Now, let's take some examples of these polysaccharides. And the most important storage polysaccharide is starch in plant cells. In animal cells is glycogen. We will soon come to glycogen as an important storage polysaccharide in animals. Now, starch contains two types of glucose polymers, which is amylose and amylopectin. Amylose is unbranched. So you see amylose here is unbranched. You have only the glycosidic bonds between the glucose molecules as the monosaccharide units forming amylose. However, in amylopectin, you have some. So in amylopectin, you have, apart from the glycosidic bonds between the glucose monosaccharide units, you also have the alpha 1,6 bond. So you see on carbon number 6, you have the bonding between the hydroxyl group, which is on carbon number 1 of another chain, and the hydroxyl group of the CH2OH, which is on carbon number 6. So you see, this is carbon number 6, as you can see in the amylose the amylose chain. But in amylopectin, you have this chain, all right, but then you have this branching as well. So you have the branching on carbon number 6 to the carbon number 1 of another monosaccharide unit in another chain. And in starch, you see this branching occurring every 24 to 30 residues. So imagine this is an amylose chain, and you start counting 1, 2, 3, 4, and you continue 5 all the way. When you reach the 24th to 30th residue, you will observe this branching point. This branching point is observed every 24 to 30 residues, and this is the alpha 1,6 branch point. Now, glycogen. Glycogen is the main storage polysaccharide of animal cells. Now, the difference, one major difference you notice between glycogen and starch, which we just saw, is that the branch point which you find in starch occurring every 24 to every 30 residue, you find in glycogen occurring every 8 to 12 residues. And what that means is that glycogen becomes more branched and glycogen is more compact than starch, and thus glycogen will store much more energy than starch. Now, glycogen is abundant in the liver as granules in the cells of the liver, the hepatocytes. And these glycogen in the liver cells break down during times of fasting to give us energy. So in the night when we sleep, glycogen breaks down into a monosaccharides, glucose, to give us energy, especially to our brains. In the skeletal muscles, you have glycogen also giving quick energy in exercise. When you are doing some exercise, some very excessive activity, you have glycogen being broken down in the skeletal muscles to give us quick energy. Now, another polysaccharide is dextran, which is bacterial and yeast polysaccharide. And apart from the alpha 1,4 bonds, which are the glycosidic bonds we saw earlier in disaccharides, in starch, and in glycogen, you also have the alpha 1,3, and you have the alpha 1,6 as well as the branch point that we saw earlier in amylopectin. So in dextran, you have the alpha 1,3, even alpha 1,2, alpha 1,4. I will urge you to draw this. You draw the structure of the glucose molecules, and once you know the carbon numbers, as you number the carbons, you try to see how the bond between the carbons number 1 and the carbon number 3 of another glucose molecule will look like, and how the bond between carbon number 1 of one molecule and carbon number 2 of another glucose molecule will be looking like. We saw that of the alpha 1,4, which is the glycosidic bond. We saw that of the alpha 1,6, which was the branch point in amylopectin. These other ones, which are the alpha 1,3, alpha 1,2, will be similar. Once you number the carbons, you will be able to identify how these bonds look like. Now, we have dextrans occurring in the dental plaque on the surface of teeth. And this polysaccharide can also be synthesized and used in several commercial products that serve in the separation of proteins. In medicine, you have dextrans being used as an antithrombotic, for the prevention of vascular thrombosis, prevention of blood clotting in some micro surgeries. They are also used as lubricants in some eye drops. Now, cellulose. Cellulose is a fibrous, tough, water-insoluble substance found in the cell walls of plants. In fact, they form the woody part of plants. Cellulose also contains glucose units, and it is unbranched homopolysaccharide consisting of 10,000 to about 15,000 glucose units. Now, it's interesting that even though cellulose contains monosaccharides of glucose, wood does not give energy to animals. If you consume wood, if wood is ingested, wood just comes out as roughage. Why? And this is because the glycosidic bonds between the glucose units in cellulose are bonded together by the beta-1,4 glycosidic bonds. Beta-1,4 glycosidic bonds. In starch, in glycogen, you see you see these bonds are alpha-1,4 bonds. Now, our enzymes, amylase, and the enzymes that break down carbohydrates in virtually all animals are specific to the alpha-1,4 glycosidic bond. So they can act on alpha-1,4 glycosidic bonds, but these enzymes are not able to hydrolyze the beta-1,4 glycosidic bonds. Even if you see termites, for instance, that readily digest cellulose, it is not because the termites have the ability to secrete the enzyme for hydrolyzing these beta-1,4 glycosidic bonds, but rather it's because in the intestinal tract of termites there is this microorganism that is having a symbiotic relationship with termites, and this microorganism, Trichonympha, secretes the enzyme cellulase, which hydrolyzes the beta-1,4 linkage. Now, the last of the polysaccharide examples is chitin, which is found in the exoskeleton of arthropods, insects, and then lobsters and crabs. This polysaccharide, chitin, is made up of the derivative N-acetylglucosamine that we saw earlier. So we have N-acetylglucosamine being the monosaccharide unit of chitin. Again here, these monosaccharide units are bonded together by the beta-1,4 linkages, and thus if ingested by vertebrates, chitin cannot be digested by vertebrates to give vertebrates energy. And on this note, I thank you for your kind attention and look forward to seeing you in the next session.